When most people hear the word cholesterol, they immediately think of something bad, something clogging their arteries or something they need to eliminate completely. But cholesterol is actually one of the most misunderstood molecules in human biology. In reality, cholesterol is essential for life. Without it, your cells couldn't function properly. Your hormones couldn't be produced and your brain wouldn't work the way it does. The real issue is not cholesterol itself, but imbalance. And that's what we're going to unpack step by step. Cholesterol is a waxy, fat-like substance that your body makes naturally, primarily in the liver. In fact, about 70 to 80% of the cholesterol in your body is produced internally, not from food. Every single cell membrane in your body contains cholesterol. It helps keep cell walls flexible but stable, allowing nutrients and signals to move in and out of cells efficiently. This is especially important in tissues with high signaling demands like the brain. Your brain alone contains roughly 20 to 25% of the body's total cholesterol. That cholesterol is critical for synapse formation, neuron communication, and overall cognitive function. Cholesterol is also the raw material your body uses to make steroid hormones such as estrogen, progesterone, testosterone, cortisol, and vitamin D. Even bile acids, which help you digest fats and absorb fat-soluble vitamins like A, D, E, and K, are made from cholesterol. So if cholesterol is so important, why does it have such a bad reputation? The problem starts with how cholesterol is transported in the blood. Cholesterol doesn't dissolve in water, and your blood is mostly water. To move cholesterol around, your body packages it into particles called lipoproteins. These particles are what we commonly refer to as LDL and HDL. LDL, or low-density lipoprotein, carries cholesterol from the liver to tissues that need it. HDL, or high-density lipoprotein, helps transport excess cholesterol back to the liver for recycling or removal. Cholesterol itself is not good or bad. It's the number, size, and behavior of these lipoproteins combined with inflammation and metabolic health that determines risk. One of the biggest misconceptions is that LDL is simply bad cholesterol. LDL particles are doing an essential job. The problem occurs when there are too many LDL particles, especially small, dense ones circulating in the blood for long periods of time. When these particles interact with damaged arterial walls, particularly in an environment of chronic inflammation and oxidative stress, they can become modified and trigger an immune response. That's where plaque formation begins. This is why balance is so important. You want enough cholesterol to support hormones, brain health, and cellular integrity, but not so much circulating in a dysfunctional metabolic environment that it contributes to vascular damage. Context matters. Someone with excellent insulin sensitivity, low inflammation, and healthy blood vessels can tolerate cholesterol levels very differently than someone with metabolic syndrome or type 2. Diabetes. Diet plays a role, but not always in the way people expect. For most individuals, dietary cholesterol has a relatively small impact on blood cholesterol compared to factors like insulin resistance. Excess refined carbohydrates, chronic stress, poor sleep, and lack of physical activity. When insulin levels are chronically elevated, the liver tends to produce more lipoproteins, particularly triglyceride-rich particles, which shifts the entire lipid profile in an unfavorable direction. This is why simply lowering cholesterol numbers without addressing the underlying biology often misses the point. Cholesterol imbalance is usually a signal, not the root cause. It's telling you something about metabolic health, inflammation, and how your body is processing energy. In this video, we're going to dive deeper into how cholesterol works in the body, what the different markers really mean, and why focusing on balance rather than fear is key for long-term cardiovascular and brain health. To really understand cholesterol balance, we need to move beyond the basic LDL and HDL labels and talk about what's actually happening at a deeper biological level. Cholesterol in the bloodstream doesn't float around on its own. It's carried inside lipoproteins, and these particles vary in size, density, and behavior. Two people can have the same LDL cholesterol number, yet very different cardiovascular risk. Depending on the number and type of LDL particles they have, LDL particles come in different sizes. Larger, more buoyant LDL particles are generally less problematic, while small, dense LDL particles are more likely to penetrate the arterial wall and become oxidized. Oxidized LDL is what really drives inflammation in the arteries. Once LDL becomes oxidized, the immune system recognizes it as a threat. Macrophages move in, ingest the oxidized LDL, and eventually turn into foam cells. Over time, this process contributes to plaque buildup. What determines whether LDL becomes small and dense? A major factor is insulin resistance. When someone consumes a diet high in refined carbohydrates and added sugars, especially in the context of low physical activity, insulin levels remain chronically elevated. This changes how the liver packages lipids, increasing triglyceride-rich lipoproteins and promoting the formation of small, dense LDL particles. This is one of the reasons triglyceride levels are such an important marker to pay attention to. HDL is often called good cholesterol, but again, it's not cholesterol that's good or bad, it's its function. HDL particles play a key role in reverse cholesterol transport, which is the process of moving excess cholesterol out of peripheral tissues and arteries back to the liver. But HDL also has antioxidant and anti-inflammatory properties. Healthy, functional HDL helps prevent LDL oxidation and supports vascular health. However, simply having a high HDL number doesn't always guarantee protection. In certain inflammatory or metabolic conditions, HDL can become dysfunctional and lose its protective effects. This highlights why looking at cholesterol in isolation can be misleading. It's the overall metabolic environment that determines whether these particles are doing their jobs properly. Another critical factor in cholesterol balance is inflammation. Chronic low-grade inflammation damages the endothelial lining of blood vessels. When the endothelium is healthy, it acts as a protective barrier. But when it's damaged by inflammation, smoking, high blood sugar, or oxidative stress, LDL particles are more likely to get trapped in the arterial wall. So this is where the atherosclerotic process begins. Oxidative stress also plays a major role. LDL particles are particularly vulnerable to oxidation when antioxidant defenses are low. Nutrient deficiencies, especially in antioxidants like vitamin C, vitamin E, and polyphenols from plant foods, can increase susceptibility to oxidative damage. This doesn't mean megadosing supplements is the solution, but it does highlight the importance of a nutrient-dense diet. Cholesterol balance is also influenced by genetics. Some individuals naturally produce more cholesterol or clear LDL particles more slowly due to genetic variations. This doesn't mean lifestyle doesn't matter, but it does mean that personalized strategies are important. For example, someone with a genetic tendency toward higher LDL may benefit even more from optimizing inflammation, insulin sensitivity, sleep quality, and micronutrient intake. Physical activity is another powerful regulator of cholesterol balance. Regular exercise increases LDL receptor activity in the liver, which helps clear LDL particles from the bloodstream more efficiently. Exercise also improves insulin sensitivity and increases HDL functionality. Even moderate amounts of consistent movement can shift lipid profiles in a favorable direction. What all of this shows is that cholesterol imbalance is rarely caused by a single factor. It's the result of interactions between diet, metabolism, inflammation, genetics, and lifestyle. When these systems are working together, cholesterol supports health. When they're out of balance, cholesterol becomes part of a larger problem. When it comes to cholesterol, diet is often the first thing people focus on. But the relationship between what you eat and what shows up in your blood is more nuanced than most headlines suggest. Cholesterol metabolism is heavily influenced by nutrient quality, macronutrient balance, and overall metabolic health, not just total fat or cholesterol intake. One of the most important dietary factors affecting cholesterol balance is the quality of fats you consume. Fats are not all the same. Polyunsaturated and monounsaturated fats, particularly omega-3 fatty acids from fatty fish like salmon, sardines, and mackerel, play a powerful role in reducing inflammation and improving lipid particle behavior. Omega-3s help lower triglycerides, reduce the number of small, dense LDL particles, and support healthier HDL function. They also directly influence gene expression related to lipid metabolism and inflammation. In contrast, diets high in refined vegetable oils that are easily oxidized, especially when repeatedly heated, can increase oxidative stress. Oxidized fats contribute to systemic inflammation, which, as we discussed earlier, creates the conditions where cholesterol becomes harmful. This is why the context in which fats are consumed matters, including food processing and cooking methods. Carbohydrates also have a major influence on cholesterol balance. Diets high in refined carbohydrates and added sugars tend to increase triglycerides and lower HDL while promoting insulin resistance. This combination strongly favors the production of small, dense LDL particles. On the other hand, carbohydrates from whole, fiber-rich foods like vegetables, legumes, and some fruits slow glucose absorption, improve insulin sensitivity, and support healthier lipid profiles. Fiber deserves special attention. Soluble fiber found in foods like oats, beans, lentils, flax seeds, and certain fruits binds bile acids in the gut. Since bile acids are made from cholesterol, this process increases the demand for cholesterol in the liver, encouraging the body to pull more LDL particles out of circulation. This is one of the most reliable dietary ways to improve cholesterol balance without medication. Protein intake and source also matter. Diets that include adequate high-quality protein help stabilize blood sugar and support lean muscle mass, which improves insulin sensitivity. Protein sources rich in micronutrients such as fish, eggs, and fermented dairy contribute nutrients like choline, vitamin B1, 2, and fat-soluble vitamins that support lipid transport and liver function. Micronutrients play a surprisingly important role in cholesterol metabolism. Magnesium, for example, is involved in hundreds of enzymatic reactions, including those related to glucose and lipid metabolism. Vitamin D, which is synthesized from cholesterol in the skin, also influences inflammation and cardiovascular risk. Low vitamin D status is often associated with unfavorable lipid profiles. And though this relationship is complex and bidirectional, polyphenols from plant foods such as berries, olive oil, green tea, and dark chocolate have antioxidant and anti-inflammatory effects that protect LDL particles from oxidation. These compounds don't necessarily lower cholesterol numbers dramatically, but they improve cholesterol quality, which may be even more important. It's also critical to talk about dietary cholesterol itself. For most people, consuming cholesterol-rich foods like eggs does not significantly raise blood cholesterol. The body adjusts its own production in response to intake. However, individuals with insulin resistance or certain genetic traits may respond differently, which again highlights the importance of personalization rather than blanket rules. Meal timing and overall energy balance also influence cholesterol. Chronic overconsumption, regardless of macronutrient composition, drives lipid production in the liver. Periods of energy balance or mild caloric restriction can improve lipid markers by reducing liver fat and improving insulin sensitivity. Even practices like time-restricted eating can have beneficial effects on cholesterol metabolism for some individuals. Ultimately, diet shapes the internal environment in which cholesterol operates. A nutrient-dense, anti-inflammatory eating pattern supports cholesterol's essential roles while minimizing its potential to contribute to disease. In the next part, we'll explore lifestyle factors beyond diet, including sleep, stress, and physical activity, and how they dramatically influence cholesterol balance. Beyond diet, lifestyle factors play an enormous role in determining whether cholesterol supports health or contributes to disease. Sleep, stress, physical activity, and environmental exposures all influence lipid metabolism, inflammation, and vascular function. These factors often don't get the same attention as food, but in many cases, they are just as powerful. Sleep is one of the most underappreciated regulators of cholesterol balance. During deep sleep, the body performs critical repair processes, including hormonal regulation and metabolic reset. Chronic sleep deprivation increases cortisol levels and worsens insulin resistance, both of which drive unfavorable lipid changes. Studies consistently show that poor sleep is associated with higher LDL, higher triglycerides, and lower HDL. Even 1 week of insufficient sleep can measurably impair glucose and lipid metabolism. Sleep also affects circadian rhythm, which directly regulates liver function. The liver produces and clears cholesterol on a daily cycle when circadian rhythms are disrupted by late night eating, shift work, or irregular sleep schedules. Cholesterol synthesis and clearance can become dysregulated. This is one reason why consistent sleep timing and sufficient sleep duration are so important for cardiovascular health. Chronic psychological stress has a similar effect. When the body is under constant stress, cortisol and adrenaline remain elevated. These hormones signal the liver to release more glucose and lipids into the bloodstream to meet perceived energy demands. Over time, this leads to higher triglycerides, increased LDL particle numbers, and greater inflammation. Stress also increases oxidative stress, making LDL particles more likely to become damaged. Physical activity is one of the most effective tools for restoring cholesterol balance. Exercise increases the expression of LDL receptors in the liver, allowing LDL particles to be cleared more efficiently from the blood. It also improves insulin sensitivity in muscle tissue, which reduces the liver's need to produce excess lipoproteins. Both aerobic exercise and resistance training are beneficial, and the combination appears to be especially powerful. Movement throughout the day matters too. Prolonged sitting impairs lipid metabolism, even in people who exercise regularly. Short bouts of walking or light activity help activate enzymes like lipoprotein lipase, which clears triglycerides from the bloodstream. This highlights the importance of daily movement, not just structured workouts. Body composition is another key factor. Excess visceral fat, particularly around the abdomen, is metabolically active and releases inflammatory cytokines. This inflammation interferes with normal cholesterol handling and increases cardiovascular risk. Losing even a modest amount of visceral fat can significantly improve lipid profiles, especially triglycerides and LDL particle size. Environmental exposures also play a role. Certain pollutants, endocrine disrupting chemicals, and heavy metals can interfere with lipid metabolism and increase oxidative stress. While it's impossible to eliminate all exposures, strategies like eating antioxidant-rich foods, supporting detoxification through adequate protein and micronutrient intake, and minimizing exposure to ultra-processed foods can help reduce the burden. Alcohol deserves special mention. In small amounts, alcohol may increase HDL, but regular or excessive consumption raises triglycerides and increases liver fat, which worsens cholesterol balance. Individual tolerance varies widely, and for some people, reducing or eliminating alcohol leads to dramatic improvements in lipid markers. What's important to understand is that cholesterol imbalance is often the cumulative result of many small lifestyle stressors rather than a single cause. When sleep is poor, stress is high, movement is low, and diet quality slips. Cholesterol reflects that internal state. Now that we've explored how cholesterol functions in the body and what influences its balance, the final step is understanding how to assess it intelligently and think about long-term health rather than chasing single numbers. Cholesterol markers are tools, not verdicts. Their value comes from interpretation and context. Most standard lipid panels include total cholesterol, LDL cholesterol, HDL cholesterol, and triglycerides. While these markers provide useful information, they don't tell the full story. Total cholesterol, for example, is a broad measure that doesn't distinguish between different lipoprotein particles. Two people can have the same total cholesterol with very different risk profiles. Triglycerides are often one of the most informative markers, and elevated triglycerides usually signal insulin resistance, excess carbohydrate intake, or liver fat accumulation. Low triglycerides, especially when combined with healthy HDL levels, generally reflect good metabolic health. The triglyceride to HDL ratio is a simple but powerful indicator of insulin sensitivity and cardiovascular risk. LDL cholesterol deserves a more nuanced interpretation. Higher LDL cholesterol does not automatically mean higher risk, particularly in individuals with low inflammation, good insulin sensitivity, and healthy lifestyles. However, persistently elevated LDL in the presence of high triglycerides, low HDL, and markers of inflammation paints a very different picture. This is why advanced markers such as LDL particle number or apolipoprotein B can provide deeper insight as they reflect how many atherogenic particles are actually circulating. Inflammation markers add another layer of context. C-reactive protein, for example, can help indicate whether the vascular environment is inflamed. Cholesterol particles are far more likely to contribute to plaque formation when inflammation is present. Without inflammation, cholesterol is much less likely to become problematic. It's also important to remember that cholesterol changes slowly. Meaningful improvements reflect sustained changes in diet, sleep, movement, and stress management over weeks and months, not days. This is why extreme or restrictive approaches often fail long-term. They may temporarily lower a number, but they don't create a resilient metabolic system. Medications like statins are sometimes necessary and can be life-saving for high-risk individuals. They reduce cholesterol synthesis in the liver and lower cardiovascular event risk in certain populations. However, they are not a substitute for lifestyle. Even when medication is used, diet quality, physical activity, sleep, and micronutrient status still matter for overall health and quality of life. One of the most important mindset shifts is moving away from fear. Cholesterol is not an enemy. It is a vital molecule that reflects how your body is responding to its environment. When cholesterol is out of balance, it's an opportunity to look deeper at metabolism, inflammation, and lifestyle patterns. Long-term cholesterol balance is about supporting the systems that regulate it: a healthy liver, responsive insulin signaling, low chronic inflammation, and resilient blood vessels. This means eating nutrient-dense foods, prioritizing sleep, managing stress, staying physically active, and maintaining a healthy body composition. When these foundations are in place, cholesterol often finds its balance naturally. And when it doesn't, you have the context and knowledge to make informed, personalized decisions rather than reacting to a single lab value. Understanding cholesterol in this way empowers you to focus on long-term health, cognitive function, and longevity rather than short-term fixes. Balance, not elimination, is the key to using cholesterol as the powerful biological tool. It was designed to be